This video is a part of the series about RNA-seq data preprocessing using the geneXplain platform. We discuss gene quantification in this video, and you can find links to other parts in the description box. After raw RNA-seq reads are mapped onto the reference, it is possible to count the number of reads assigned to specific genomic features. This process is called read summarization or read quantification, and it is required by a number of analyses, such as gene expression analysis, for example. The output of quantification is a count table, in which the number of reads assigned to each feature of interest is recorded. A feature can be exons, genes, or transcripts. A general feature format, GFF, describes genomic features in a tab-delimited text. The format has several versions, but there are usually fields describing the feature and its genomic position. Most tools work best when counting uniquely mapped reads at gene level. Each gene is then considered as the union of all its exons, and total read count for a gene is the sum of reads associated with each of the exons. There might be a situation when a read maps to more than one location in the reference annotation. Such reads are called multi-mapping reads and ignored by default by many tools. It is recommended to not count multi-mapping reads. Any single fragment must originate from only one of the target genes, but the true origin cannot be determined for such cases. Here's an illustration of different counting modes for Hdsec count quantification tool. There are options to count an ambiguous read for both genes. However, such options should be carefully used, as they may lead to overcounting and later problems with counts normalization. There are several tools available under Counting Reads to Genomic Features group of RNA-seq preprocessing in the geneXplain platform. Again, as with the alignment tools, the core parameters are discussed for feature counts only, but the comments are relevant for other quantification tools as well. The input is a track file with aligned reads in either SAM or BAM format from the data tree. We discussed BAM files in the video about read mapping. The next input is a GTF annotation. geneXplain platform includes inbuilt annotations to be used with the reference genomes provided for alignment methods. Please be sure to use the same reference for mapping and counting steps to receive meaningful outputs. It is also possible to provide a custom annotation file from a data tree. Additional parameters are required for a custom annotation. Feature type specifies which value to take from the third column of GTF as feature, and attribute type specifies what attribute to extract as a meta feature. The default values, also used with inbuilt annotations, are exon and gene ID respectively. We use inbuilt ensemble annotation for human genome. Read mapping results usually include mapping quality scores for mapped reads. The minimum mapping quality parameter allows to skip reads with alignment quality lower than the given value. Counting methods support strand-specific read counting if strand-specific information is provided. For paired end reads, the strand of the first read is taken. The full parameter list contains some advanced parameters. Count at feature level parameter allows to count exons or other features according to the annotation provided before. In the case of paired end reads, two reads define one fragment of interest, and feature counts is instructed to count fragments rather than individual reads. It is recommended to use the same values for minimum-maximum fragments lengths as during the alignment step. Although the output path was set automatically, let's give the resulting files more meaningful names. There are several settings to regulate counting of multi-mapping reads, as we discussed earlier. Please consult the info box before changing these settings. The analysis can be started after that. The resulting files should appear in the data tree upon completion. Open the log file to check the overall performance of the counting. The summary table includes more specific quantification statistics. The main result is the count table. Usually, count tables have two columns, a column with features and a column with the corresponding counts. In this example, the features were genes, so we have gene ID column and the last column called count that shows how many reads map to a specific gene. The feature counts program also annotated the count table with the additional data, which might be necessary at later analysis. Please note that raw read counts require normalization before expression level comparison. The differences in gene lengths or in total number of reads per sample can lead to skewed results. Some quantification programs report counts already in a transformed way. Metrics like reads per kilobase million, fragments per kilobase million, or transcripts per million try to rescale the counts to correct for library size and gene lengths. However, it was recently shown that application of normalization methods to raw counts provide more accurate account of the expression levels. We will not go into detail about these methods, but recommend to explore the topic more for performing better quality analysis. The geneXplain platform provides many tools and workflows to work with the RNA-seq data. Check other videos on this channel to get ideas on how to go further with your count tables. Thank you for watching.